Welcome, everyone. As Nicole said, I am Heather and I go by Heather. Um, and a little bit of background. I have an undergraduate degree in math, a PhD in immunology and microbiology from the University of Colorado Health Sciences Center in Denver. And then I did a postdoctoral fellowship and taught medical school at Yale University before coming to NUNM many, many moons ago. My current research is on Parkinson's and the gut-brain axis. And so what we thought we would talk about today is a little bit on the gut-brain axis. It's pretty scientific. So if science isn't your thing, I'm going to try to break it down for you. But we're going to go through pretty slowly on the gut-brain axis, and we'll end with some stuff on Parkinson's. So with that as an intro, let's go ahead and start. All right, so we're starting with the gut. And as we think about the gut, we usually think about it as this mushy little organ that sits right below our diaphragm that hurts when we eat bad food. But the reality is, as we think about the gut, we need to be thinking about the microorganisms inside the gut, because your gut is chock full of microbes, and those microbes are producing things that are affecting your entire body. So as we think about microbes, usually we just think bacteria, but the reality is in the gut, we're talking about bacteria, viruses, fungi, and protozoans. They all live in your gut. Now, for the interest of time, I'm just going to talk about the bacteria today. I teach a class at NUNM called the gut microbiome, where we talk a little bit more about other things as well, like the viruses and the fungi and the protozoans. But today we're focused on bacteria. The bacteria in your gut are 90% of the microbes that you have in your gut. So 90% is bacteria. The other 10% are the viruses, the protozoan, and the fungi. Of the bacteria that are in your gut, there are two primary phyla. They're called Bacteroides and Firmicutes. And the reason that you should know these is that if you are doing a test to test how healthy your gut is, they will do a ratio of Firmicutes to Bacteroides. Now, is there an optimal ratio? I don't know that there is. What Genova, a big company that studies this, is saying is that usually you want 12 Firmicutes to 620 Bacteroides. Here's the thing. When people say that there's an optimal ratio, usually the way that they're doing it is that they're studying a typical person or a typical profile. And because our gut is very much influenced by the food we eat, I don't know that I can say that one person is more typical than another. And so is that truly optimal? Yeah, I'm not sure, but you will see in the literature, you'll see Firmicutes to Bacteroides ratios being used as a measure of health. Your ratio of Firmicutes to Bacteroides is going to differ depending on where you live. In the United States, you'll see that we have a whole lot of Bacteroides and we have a lot of Firmicutes. We don't have very many actinobacteria, but look over here at Japan. Japan, where they eat a lot more fish, has a lot more of the actinobacteria. And you'll see that like in Sweden, we see more archaea. So we see different types of bacteria depending on where people come from because their diets are different. Why do we have so many Bacteroides in the United States? Well, it's because we're red meat eaters, and we see more red meat is associated with more Bacteroides. So you'll also see that another species of bacteria here is a Prevotella, and that's associated with eating sugar. So here we are with our American diet, with our red meat and our sugar, and we see a lot of Bacteroides. You actually have many, many microbiomes around your body. You not only have a gut microbiome, you have a hair microbiome. Your scalp has its own microbiome. Your mouth has its own microbiome. Your nostrils have a microbiome that is different than your mouth. partly because your nostrils are colder than your mouth. So you need to have microbes in your nose that can withstand cold, especially if you live in Minnesota. I just got back from Minnesota and it is darn cold. Lots of cold loving microbes there. Then we have microbes in our stomach. We have microbes on our skin, in our vagina, if we have a vagina, in our colon and our gut. So lots and lots of different microbiomes. And the thing to remember here is that they all interact with each other. So you may have something that's going on in your skin that's actually being triggered by your mouth. Or you might have something that's going on in your hair that's being triggered by your gut. So the fact that all the microbiomes are interacting with each other is something we're only really starting to understand how to control. There are so many things that we are exposed to day in and day out that change the composition and the function of our microbiomes. So our genetics have something to do with our microbiome, but more than our genetics is our diet. Our diet has a lot to do with our microbiome. Our age has to do with our microbiome. Our microbiome is seeded when we are born. And so whether you are born vaginally or C-section is also going to have to do with your microbiome. And then whether you're breastfed or formula fed has to do with your microbiome and how you are going to live the rest of your life. You might not know that the way that you were born and whether you were breastfed or formula fed has that effect, but it actually does. The number of courses of antibiotics that you have had in your life and the timing of those antibiotics has an amazing effect on your microbiome. And there was a study out of Germany a couple of years ago that shows that non-antibiotic medications also affect your microbiome. Whether or not you exercise, whether or not you're a smoker, and then, as I showed you already, your geographical location. I could take this slide and I could make 30 more things on this slide that have an effect on your microbiome, like whether or not you have pets and whether or not you live in the country or live in the city, whether you drive a diesel car or a gas car. All of these things are affecting your microbiome. And that's an important distinction because when things start to go wrong, we always try to pinpoint it to one thing, and it might be a really complex combination of things that are happening there. All right, what are the basic functions of the bacteria in your microbiome? Number one thing is to protect you from pathogens. Keep in mind that usually when you encounter a pathogen, it's coming through your mouth. So the very first microbes that encounter a pathogen are your oral microbiome. And then often you're swallowing things, right? You eat every day. And so those microbes are going to go down to your gut. And as a result of that, you have antimicrobial peptides that are produced by the microbes in your gut to keep those other microbes that might be pathogenic at bay. The colonization of the microbes in your gut also try to squeeze out any pathogenic microbes that you might be exposed to in your daily living. Your microbes are responsible for digestion and metabolism. So we know from experiments where they have done fecal transplant, and yes, that is what it sounds like, where they transplant the fecal matter from one mouse to another mouse or one human to another human, that our metabolism is primarily a result of our microbiome. It's not how many calories we eat, as it turns out, it's which microbes we have to digest those calories. So we can take the microbiome of someone who is thin, put it in someone who is chubby, and they will become thin. Pretty interesting. They control epithelial cell proliferation and differentiation, especially in the skin and the gut. So if you have issues with eczema on your skin, likely that's a microbiome imbalance. Likewise, if you have issues with IBS or inflammatory bowel disease or Crohn's disease in your gut, often that's a microbiome imbalance. They modify insulin resistance because they affect insulin secretion. So for people who have issues around insulin resistance, like type 2 diabetics, it's a microbiome issue. And then what we'll talk about today is they influence the brain-gut communication. So they affect mental and neurological functions, not only your mood, whether or not you're depressed or anxious or happy or attentive or motivated. It's all a microbiome thing. So you can blame it on your microbiome. All right. Bacteria exist in ecosystems. So let's just review what an ecosystem is. It's a community of organisms together with their physical environment, and the organisms can be different in different ecosystems. So I like to think about this as think about a marine ecosystem with all the fish and the plankton and the coral, and all of those things come together to form an ecosystem. But you could also have a desert ecosystem, which has cacti and roadrunners and Wile E. Coyote, right? Or you could have a forest ecosystem. So all sorts of different kinds of trees. They're all ecosystems. They're all healthy. Those broad categories of ecosystems are called biomes. In the ecosystem, both matter and energy are conserved, which means that energy is going to flow through the system, usually from light to heat, while matter is recycled. And if you think about how matter is recycled in your body, your microbiome is recycling food into waste products. Ecosystems with higher biodiversity tend to be more stable, with greater resistance and resilience in the face of disturbances. So what we see with the gut microbiome is the more diversity that you have in your gut microbiome, the healthier you are. How that translates is the more diverse the foods you eat, the more diverse the microbes you will have, and the more resilience you will have in your microbiome. So if you are in the habit of eating the same 10 foods every week, you do not have a diverse microbiome, and that is not healthy, even if they're healthy foods you're eating. What your job is to get a diverse set of nutrients to feed the different microbes that are in your microbiome. Now, also in your microbiome, we have something called a keystone species. A keystone species is an organism that helps define the entire ecosystem. And the big thing about a keystone species is that they are not functionally redundant, meaning that if one species disappears, the entire ecosystem collapses. No other ecosystem is able to fill its niche. This is a concept that's really well illustrated through the wolves of Yellowstone. So, you know, in the 1920s, the government allowed the extermination of the gray wolf in Yellowstone Park. And when that happened, a lot of different species no longer had a predator to thin the crowd. So you saw an overabundance of elk and an overabundance of coyotes and beavers and bears. As a result of that, especially because of the beavers, we saw a different way that rivers would flow. And then we saw different animals that lived along the banks of the rivers. When the wolf was reintroduced in 1995, it completely changed the entirety of Yellowstone Park, where now we see very different birds that nest along rivers. Rivers have rerouted themselves again because now beavers, are not around to change the way that the river flows. There's different fish that exist, etc. The wolf is a keystone species for Yellowstone. If I were to wipe out the beavers in Yellowstone, I would see a very different thing than when I wipe out the wolves in Yellowstone. So think about that when you think about the fact that we take antibiotics. And if we have an antibiotic that wipes out a keystone species, it can make us really, really sick. Within your gut microbiome, you have 3 different enterotypes. Entero is the prefix that we use for the gut. So when you see entero, you're going to think gut. These 3 enterotypes are Ruminococcus, Prevotella, and Bacteroides. And if we look again at where different enterotypes exist, we see that as we go through different places, like Argentina, we see way more of the Bacteroides, USA, Bacteroides, and here we have our Firmicutes. But these different enterotypes are different in different locations because of the different foods we eat. So which enterotypes are with which foods? Bacteroides is primarily red meat. So when we see Bacteroides, that is people who are big time red meat eaters. Ruminococcus, think ruminants, think cows, right? These are people who eat vegetables. Lots of vegetables is ruminococcus. And Prevotella, well, those are the people who love sugar and sweets and carbs. So I broke it down for you here. Ruminococcus, we think resistant starches, we think vegetables, potatoes, carrots. One of the big things that ruminococcus can do is they can degrade mucus. And so in your gut, they will help keep mucus degraded so that things will flow through easier, which is why we give people particular foods that are starches to treat constipation, as an example. Prevotella is carbohydrates and simple sugars. And Bacteroides, animal fat, high protein. Now, as we keep going, we're going to talk about some famous gut bacteria. So these are bacteria that you're probably going to recognize, or if you don't recognize, you probably should recognize somewhere in your academic life cycle. E. coli, Escherichia coli, produces vitamin K2. And when E. coli is out of balance, when you make too much of it, you get food poisoning and diarrhea. But E. coli is a normal part of your gut microbiome. Lactobacillus casei, casein, dairy. So this is one of the subspecies of lactobacillus, lacto, dairy, casei, dairy, and this produces vitamin B1 from dairy. Ruminococcus, I already just mentioned that one. That was one of the first species that was discovered. And what we see is that ruminococcus tends to be low in people who have inflammatory bowel disease or Parkinson's. Akkermansia. Akkermansia is getting a lot of attention these days because it has been shown to help with glucose metabolism. And people who lack Akkermansia tend to be more obese and they tend to age faster. So let's talk about the story of Akkermansia for a second. In humans, if they have Akkermansia in their normal microbiome, we see that they have a reduced weight circumference by 1.5 centimeters, which isn't huge. However, we also see a decrease in inflammation, and we see that people who don't have Akkermansia tend to be more obese. They tend to have diabetes, they might abuse alcohol, and they have more intestinal inflammation. So why is Akkermansia getting so much attention? Because they have now invented Akkermansia probiotic. And people thought, oh boy, if this is true, if people who are thin have more Akkermansia and people who are chubby have less Akkermansia, then all we need to do is give these folks who are overweight Akkermansia and they're going to lose weight. Do you think it works? Of course it doesn't work. If it were that easy, this would have been solved a long time ago. But a lot of people still believe it. And so Akkermansia is one of the most popular probiotics on the market right now. Another bacteria that you're going to recognize from probiotics is Bifidobacterium. Bifidobacterium is one of the first species to populate you when you are an infant. Its job is to ferment breast milk. So it actually is populated both from breast milk and from the nipple. This is why children who are breastfed have a different microbiome than children who are not breastfed. If you're breastfed, you have Bifidobacterium. What's really important about Bifidobacterium, it is one of the most anti-inflammatory microbes that we have. In adults, Bifidobacterium ferments plants that we eat, so carrots and spinach and all of those fun things. And people who don't eat vegetables tend to have lower levels of Bifidobacterium, which is a problem because Bifidobacterium is anti-inflammatory. So what we see is that people who have inflammatory conditions like IBS, inflammatory bowel syndrome, or ulcerative colitis tend to have less Bifidobacterium. We see less Bifidobacterium in almost every autoimmune disease as well. So this is a time when you would want to eat Bifidobacterium, But you would also want to give the bifidobacterium the food that it wants. So either fermented plants, or one of the things that's on the market now is colostrum. And you can take colostrum as a supplement and it will feed your bifidobacterium and bring them back to life. Prevotella, I already mentioned, is our sugar loving microbe. It produces LPS, and LPS is a bacterial polysaccharide, lipopolysaccharide, which is highly inflammatory. We're going to talk about LPS a little bit later on as well. So why do you get inflammation when you eat sugar? Partly because of the Prevotella. Partly also because every time you eat sugar, sugar has to go through the Krebs cycle and you produce reactive oxygen species, which are also inflammatory. So we've got a couple different mechanisms for why sugar is so inflammatory. And then Bacteroides vulgaris. The reason that I put Bacteroides vulgaris in here is that it is able to degrade glycans. Anybody know where there are glycans in our food supply? Mushrooms. Mushrooms have glycans. And Bacteroides loves to degrade glycans and then reduce your cardiovascular disease. So what we see is that people who don't eat mushrooms have a higher incidence of cardiovascular disease. Pretty crazy, huh? It all goes together. All right. How does this work? You eat food, so you ingest dietary fiber, and that dietary fiber we could call a prebiotic. When you eat that food, your bacteria ferment it in your gut. So you can ferment it on the countertop and have fermented foods like sauerkraut or kombucha, but that's exactly what's happening in your gut. You are fermenting that food product, and you are fermenting it into something that we call a postbiotic. So the food itself is a prebiotic, the bacteria are probiotics, and what you make when you ferment those foods is a postbiotic. It is the product that is made from the bacteria. Now, as you can imagine, if you have a couple of bacteria, you're going to make a small selection of postbiotics. If you have all the bacteria that exist in your gut, which is pounds and pounds and pounds of bacteria, you make a very complex mixture of postbiotics. And we're starting to see postbiotics appear on the market as a therapeutic agent. So there are postbiotics that just come from lactobacillus, very simple postbiotics, or you have postbiotics that are derived from healthy humans, which are very complex postbiotics. Now, what's also interesting is those bacteria produce those metabolites, those postbiotics, and other bacteria use those metabolites. So that's called secondary metabolism. And then human cells and even more bacteria use those metabolites. It's like a whole domino effect, right? You eat the food and then the first bacteria comes in and then the next one and then the next one. So if you're not eating a really rich, complex diet, not only have you shut down the first pathway, you've shut down all the additional pathways that come after that. Now, another thing that we need to do is we need to keep our gut microbes in the lumen of our gut. It would not be good for us if these microbes escaped into the rest of the body. And in order to do that, we have these intestinal epithelial junctions. So this is an intestinal epithelial cell. You'll see it has this brush border on it. So this is looks like little finger-like projections on the cells in your gut. And that helps to push the digesta along when you are pooping. So these little fingers like to flow one direction so that you poop, okay? Now, what holds those cells together are these proteins here, which are junction proteins. And these are called tight junctions. And there are several different types of junction proteins that create this barrier function, because it's a really important barrier. So we've got tight junctions that are called occludins, claudins. and other junctional adhesion molecules. Then we have adherens junctions, which are similar but a little bit different. They're called cadherin and adhesion receptors. We have gap junctions, and we have desmosomes. All of these are important for keeping your intestine from allowing things to just randomly flow through. Now, sometimes we develop something that's called intestinal permeability. The layman's term for intestinal permeability is leaky gut. And what leaky gut is this, or intestinal permeability is it's when we allow things to go past the tight junctions and into the body. That can be pretty dangerous. We want some nutrients to do it, but we definitely don't want bacteria to be doing that. So there are certain things that help with our tight junctions. For example, if you eat vitamin A helps create the mucus barrier that sits along the top of the epithelial layer here. Another thing that can happen is, so here's our mucus. We get that from vitamin A. We make an antibody called IgA, or immunoglobulin A, that also helps keep these junctions intact. If these junctions aren't intact, then we get increased intestinal permeability. And either bacteria or, let me show you here what LPS might look like. It is on the extracellular cell wall of the bacteria. So either the bacteria itself will go across the junction, or the LPS from the bacteria will go across the junction. What happens? What happens if LPS makes it across that tight junction? It binds to lipoproteins, so it doesn't go around wreaking havoc or causing sepsis. The lipoproteins lower LPS in circulation. And then those lipoproteins are recycled through the liver, and that allows LPS to be recycled too. Now, interestingly, statins will increase the LDL receptor to get rid of these lipoproteins. When those LPS levels are elevated, the lipoproteins are also upregulated. And then when you have small particles of LDL, they don't recycle well at all. And so macrophages try to clear them, and that can create what's called a foam cell in your blood vessels, which can lead to atherosclerosis. So we don't like LPS anywhere other than in our intestinal lumen. And that leads us to the brain, because just like your gut has a barrier to try to keep things out of your body, your brain has a barrier called the blood-brain barrier to try to keep things out of your brain. And the blood-brain barrier is made up of those very same tight junctions that you have in your gut. Whoa, mind blown? Very same tight junctions in your gut and your brain? That seems like it could be important, because the tight junctions that you have between endothelial cells also hold the blood-brain barrier together on the basement side of the blood-brain barrier, right in here. So if you have LPS because you have leaky gut, it can disrupt the blood-brain barrier. In other words, you can have inflammation in your brain because you have inflammation in your gut. That seems important. Write that one down. Okay, let's keep going. So what's the effects of intestinal LPS on the blood-brain barrier? LPS binds to a particular type of receptor. It's called TLR4. Why? Because immunologists are dumb and they name everything with letters and numbers, which makes it really hard to remember them. What you need to understand is that when LPS binds to a cell in your brain, it gives it a danger signal, and it shifts the cells in your brain from a protective role to attack mode. In other words, you just start having inflammation in your brain. Now, a different cell type called an astrocyte moves away from the periocytes, and it's going to disrupt the blood-brain barrier, which allows a further increase in the permeability of the blood-brain barrier. And then more inflammatory molecules can get into the brain, and you wind up with neuroinflammation. Let's think about what diseases of neuroinflammation there are. Neuroinflammation is Alzheimer's, Parkinson's, ALS, multiple sclerosis, Huntington's disease, and we even think there's some neuroinflammation that happens with anxiety and depression. Autism, yeah, lots of issues here. Is there a gut-brain relationship in all of those diseases? There absolutely is. And we're just now elucidating exactly what those mechanisms are. Now, I just gave you the effects of LPS, but does it have to be LPS? No. There's actually other toxins made by other bacteria that can get across the intestinal barrier as well. And any toxin that can get into the bloodstream can also cause havoc on the blood-brain barrier. So we now know that 50% of all dementia begins with the breakdown of the blood-brain barrier and is probably coming from the gut. My stepfather has vascular dementia, which is different than Alzheimer's. With Alzheimer's, we tend to have dementia that goes with anger and hostility and some difficult emotions. Whereas vascular dementia, often people are happy, they just can't remember anything. So they have trouble with their memory, their perception, their reasoning. Today, the doctor was telling me that they were upset because my stepfather was brushing his teeth more than once in a row. Yeah, he doesn't remember that he already brushed his teeth, but is it a problem for him to brush his teeth more than once? Probably not. Now, how does LPS get out of the gut? How do you break down that gut barrier? Well, the first thing is stress. It turns out that stress can lead to the degranulation of mast cells. You're going to know a mast cell because a mast cell causes hives when it degrades in your skin. Well, when it degrades in your gut, it degrades the tight junction. Obesogenic diets can lead to LPS getting out of the gut. High fat, high sugar, low fiber diets. We see LPS increases in the blood 4 weeks later. It's 71% of people. And the question is, does it take 4 weeks? It probably doesn't. It's just that's how the study was done. So they looked at 4 weeks was their first time point. Obesity itself increases a protein called zonulin. And zonulin can lead to increased intestinal permeability. Now, zonulin's actually kind of interesting because zonulin is a protein that mimics a component of wheat. So when people say that they can't eat wheat, is it an allergy? Maybe, or maybe it's that they have this protein that looks like zonulin, which increases their intestinal permeability and makes them darn sick. And then binge drinking. When I'm saying binge drinking, I'm talking more than 3 to 4 drinks per day for women or 4 to 5 drinks per day for men. So zonulin. Zonulin looks like wheat. Gliadin, I'm from Minnesota. We pronounce it gliadin. Some people call it gliadin. Gliadin from wheat binds to a particular receptor called the CXC3 receptor, and that activates zonulin, and zonulin is going to open that tight junction. Whoops, for about 30 minutes. So when you eat wheat, it can open your tight junction for about 30 minutes. In people with celiac, it's worse. In people without celiac, the tight junction opens for less time. Does that mean that gluten's bad for everyone if gluten triggers the opening of the tight junction? Well, no, because we know that if we increase whole grains, including gluten, it reduces all-cause mortality, especially, so here's our total mortality. It reduces by 25%. But look at cardiovascular mortality. It's reduced even more. So it's not that gluten is bad for everyone. Whole grains that contain gluten are not bad if you're not sensitive to them. But if you are sensitive to them, then yes, they can be bad. Why is that? Well, there's a 2-hit hypothesis of intestinal permeability. And they've done this experiment in animals where mice that have genetic defects in their tight junction associated proteins, they don't get sick because IgA compensates for that loss of barrier function. That antibody compensates. But if you get a second hit, like stress and drinking or stress and an obesogenic diet, now we've got two hits and now we do not have the ability to compensate anymore. So how do we fix that? Well, one of the things we do is we increase a chemical, a short chain fatty acid that's called butyrate. How do you get butyrate? You eat pectins. You eat berries, citrus, and root veggies. You eat beta glucans like mushrooms or oats. You eat inulins, garlic, onion, and artichoke. And you eat resistant starches, green bananas, or potatoes that you have cooked and then let go cold in the refrigerator, and then you recook them. That makes them a resistant starch. Other ways to fix that intestinal permeability includes omega-3 fatty acids. They lead to the production of an intestine alkaline phosphatase, and that binds to a receptor on the intestinal epithelial cell membrane, which changes the pH of the gut, reduces the number of gram-negative bacteria, which means you'll have less LPS. So omega-3s and healthy foods, that's how you're going to fix your intestinal permeability. All right, we're going to wrap up with Parkinson's. So Parkinson's is one of these diseases that we know is affected by the gut-brain axis. There was a 2015 study by a group led by Sheppergen, which is Denmark. And what they did is they compared the fecal microbiome of 72 people with Parkinson's disease compared to 72 control subjects. And what they saw is that the abundance of a particular species of Prevotella was reduced in people with Parkinson's. Now, this is interesting because this is a different Prevotella than the Prevotella that we have in the United States. This is not a Prevotella associated with sugar. It's a different one, okay? Now, you can't have a gap in your gut microbiome. So if something is reduced, something else has to overgrow to fill the space. And what they saw is that when Prevotella is reduced, we need enterobacteria to overgrow to fill the space. When enterobacteria overgrows, we see postural instability and difficulty walking. What? Difficulty walking is coming from gut bacteria? Yes, it is. This happened in 2015. It has now been repeated in the United States. So we did it in the United States as well. This group did it. And one of the first things they looked at is they recruited people with Parkinson's and people who were controls. They collected stool samples. And their hypothesis was that if they did a free microbiome-wide association study, that they would see a difference between people with Parkinson's and people who were controls. Well, the first thing they looked at was different locations. So if you see in this first place, we have Parkinson's cases in the first panel. We have control cases in the second panel. And the different colors are different locations. And what they thought they were going to see is different microbes associated with Parkinson's versus controls in this panel. They didn't see that at all. What they saw is that people from Birmingham have a very different microbiome than people from Seattle, Atlanta, and Albany, New York. Okay, I already told you that geographical location makes a difference. So this showed us geographical location makes a difference. Now they looked at different microbes. Okay, so cluster one, if you look down here, you're going to see the different microbes. These are the microbes that are overgrown in people with Parkinson's in the United States. In the United States, we see that Polyphymonas is overgrown. Prevotella, which was undergrown in Denmark, is overgrown in the United States. So this is too much sugar, right? And then a Corynebacterium. Then they have 10 species. Oh, by the way, here's how you read this. In blue is the people with Parkinson's, and in orange are the controls. Blue, people with Parkinson's, orange, controls. Whoops. And so what you can see is that there are more blue microbes here with Polyphemonas, Prevotella, and Corynebacteria. Now, the next cluster, these 10 microbes, are undergrown in people with Parkinson's. In other words, you're going to look for the orange bars to be higher than the blue bars. And indeed, all of these show that the orange bars are higher than the blue bars. These are the microbes that make something called short chain fatty acids. In other words, remember when I was talking back here, butyrate? Butyrate is a short chain fatty acid. People with Parkinson's are not, they're missing the microbes. So they can eat these foods, but they're missing the microbes that make the butyrate. Oh, well, shoot, that's a problem, isn't it? So they're missing those microbes. So how do we bring those microbes back? Well, that's where you come to NUNM. And we're going to teach you how to bring the microbes back. Nutrition, you'll learn. Naturopathic medicine, you'll learn. Chinese medicine, you'll learn how to bring those microbes back. I'll give you a hint. Has a little to do with that slide I just went back to. Healthy food, healthy diet. So there you go. That's a little bit, that's an introduction to the gut-brain axis. I hope you learned something that you didn't already know today.